welcome to Lecture 2 of ZOO3649, Evolutionary Genetics. Today we're going to be talking about whole genome sequencing, okay? In the last lecture we discussed GBS, we discussed RADseq. They are reduced genome sequencing, reduced representation. Because why? Sometimes we don't have enough money to sequence the whole genome. So we can sequence a small piece of it, right? But that small piece that you sequence at random using RADseq or GBS, it still comes to thousands, hundreds of thousands of SNPs, okay? And with all that polymorphism, you are able to do population genetic studies, you're able to do a lot of interesting genetic work, a lot of interesting research, okay? But for some things, that won't do. Reduced representation won't do. For some things, you need the whole genome, okay? And today, we're going to be talking about the whole genome, sequencing the whole genome, okay? And if you are lucky enough to do honors in my lab, you will be doing work only with whole genome sequences, all right? So something to look forward to if you are clever. Let's talk about it. All right, the complete genome sequence. You know, when I was your age, I'm not that old, but when I was your age, not long ago, the idea of having the whole sequence of the genome, the idea of knowing it was like a science fiction. It was unbelievable. You couldn't think, how could we possibly sequence the whole genome using Sanger technology? But today, we have honors students analyzing whole genomes. That is how far we've come since those days, not long ago. Yeah, the whole genome, what does it contain? What sequences does it contain? It's got all the chromosomal DNA, right? From the nucleus, all the nuclear DNA. It's also got all the organelle DNA. Remember the theory of endosymbiosis. We have chloroplasts, we have mitochondria, we have organelles in eukaryotic cells, which have their own DNA. So when you smash up a cell or some tissues and you extract the DNA and you sequence it, the whole genome is not just the nuclear genome. The whole genome is also the genomes of those organelles that are in that tissue. The mitochondria, and if it's a plant, mitochondria and chloroplasts. Okay? So the organelle DNA is also part of the whole genome. Okay? So the process is actually quite straightforward. Okay? We can sequence it, whole genomes, in two different ways. And you are going to be tested on this, guys. So put your thinking caps on and remember what I'm saying. There are two ways to sequence the whole genome. The first way is the hard way, is the expensive way, but it has to be done. The first genome for anything has to be done this way. There's no way to do the first genome any other way than the way I'm telling you now. And that is what we call reference genome sequencing. So, for example, the human genome, we have now sequenced thousands of human genomes, but there's still one reference that we map all of those other genomes to. There's still one gold standard called the reference, okay? And that has to be obtained what we call de novo, okay, from scratch, okay? If there's no reference genome, you will have to establish one. You will have to sequence one before you can sequence other genomes, all right? And that is where it gets expensive, because a reference genome is not easy to establish, okay? Why? Because you need to make several different libraries, each having pieces of DNA of different sizes, small pieces, medium pieces, and very big pieces, okay? And you need all three sizes of DNA of fragments to be able to assemble a DNA, a whole genome from scratch, de novo, okay? So that's the problem, okay? And so when you sequence all of these libraries, you make it de novo, okay? You make it from scratch. De novo means new, from scratch, from nothing. Okay? And what is important here is what we call coverage. Okay? Coverage is basically how many times on average the same position in a genome gets sequenced by the Illumina sequencer sequencing a fragment. Okay? And the higher the coverage, the better the quality of your genome is. Okay? So if it's something above 50 times coverage, that means every base, every SNP, every position on that genome has been sequenced at least 50 times, on average, 50 times by the Illumina sequencer. In order to get that kind of coverage, you need to do a lot of sequencing. And that is why it gets expensive. Okay? That's why it gets expensive. The second way, okay, is the easy way. It's called resequencing. So resequencing basically means what? If you already have a reference, then you don't need to spend big money on your second genome, or your third, or your fourth, or your millionth. You don't. You just need one reference, and the next genomes, all the others, you can sequence. You don't have to sequence a lot, because you don't have to assemble them de novo. You don't. You already have a reference that you made de novo from nothing. So now when you sequence the second genome, you don't have nothing. You already have a reference. So what do you do with that? You don't do deep sequencing. You don't do a lot of sequencing. You do very light sequencing on the Illumina machine. And then you map it. You line it up, like we line up our Sanger sequences. You line up this resequenced genome with the reference that you already have, the expensive one. Now you can sequence a cheap one and just map it to because it's not de novo anymore. You've already got one. So the second one, you don't have to spend so much money. Okay? You just sequence a very little. It doesn't have to be a high coverage. Okay? And then you map it onto the reference because you already have a reference. You're not doing it de novo. Okay? And so you can sequence to lower coverage between 1 and 20x. Around 16x is about the limit for where you can call heterozygote sites. So if you you look at heterozygosity in your whole genome, the coverage of the genome has to be at least 16x, okay? Because Illumina makes a lot of mistakes. And so if you have, say, a 5x genome, that means every position is covered by 5 Illumina sequences. Now how do you know, say, one of those sequences is an A and the 4 are a T? How do you know that the A was not a mistake? How do you know if the A is actually a second allele? The one given by, the T can be given by mom and the A could have been given by dad. So that individual could be heterozygote. With only five independent sequences for that position, there's no way of telling. But if we had 16 independent sequences for that position, we will be able to tell very clearly which is a heterozygote and which is just a mistake. Okay? A mistake will be one off. You'll have one A or one T or something like that. But if it's a heterozygote and you have 16x, you will have many with the one allele and many reads with the other allele. And the mistakes, there will just be one here or there. Okay? And that's why you need a high coverage to be able to call heterozygotes, to be able to phase a whole genome sequence. If you have low coverage, your whole genome sequence is unphased. It's like Sanger sequence. Okay? Important to remember. Please don't forget that. All right. So how do we get our reference genome de novo? The one that's expensive. How do we do it? I'm going to talk you through it. Okay? We call it the hierarchical genome assembly process. Hierarchical means we just follow a process one after the other, okay? It's in a hierarchy. So first, what do we do? Here's your green is your genome, okay? That is the genome inside the animal or the plant or whatever you're interested in. You want to sequence that genome, right? At the very bottom is the blue line. That is what? That is the very same genome, but that is it sequenced. You see here on the green, there's no sequence. It's the genome it doesn't have a sequence. We want to take it from the green to the blue, where you see underneath that blue, you see the sequence there. That's what we want. Okay? So what do we do? Here it says shotgun. That basically means we restrict, chop it up into millions and millions of little pieces like this. Then what do? We put them into, we ligate them into a vector. Okay? We amplify them and we sequence them. And then what do? We have different sized molecules and we try to basically assemble them into the right place. Okay? Now, doing this de novo is very difficult because if you have nothing to start with, how do you know where each piece goes? That's the hard part. That's why you need to divide the DNA into 3 tubes. Okay? This green genomic DNA that you've extracted here, you put it into 3 tubes. Okay? The first tube, you shear, make everything a small size, the way you would do with RADseq. All right? The second tube, you do a mild shear. You don't hard shear it. You do just a very light shearing. And what will happen? The DNA sizes will not be small. They will be slightly bigger. Okay? Then what do you do in the third batch? You don't shear at all. So what is this? All the DNA pieces are quite big. Okay? So now, this is how we get tiny small pieces, medium-sized pieces, and big pieces of DNA. Remember, we want to create the reference from de novo. We need these big pieces here. Because otherwise, these little pieces don't know where to join up with each other. These big pieces and medium pieces act as a guide for that. That's why it's called a hierarchical assembly process. All right, so we take the first tube where we have this small fragments, and we put into a vector and we sequence it, right? Then the second and third batches, what do we do with that? We can't just put them because they're big pieces of DNA, maybe 100 kb, yeah, maybe 50 kb, maybe 200 kb. So what do we do with those big pieces? You can't put it into a plasmid. Now you remember why you learnt about vectors in 2544, right? Because you need to put it into a YAC vector. Because a YAC vector takes sizes which are much bigger. Remember, what size does a cosmid take? What size does a plasmid take? Yeah, that was memory. Now you know why you need to know that. In third year, if you don't know that YACs take big pieces, you're going to be struggling. You're going to try to memorize this slide. But if you knew it from the second year, it's obvious we put the big fragment into a YAC because that's the vector that can take a big piece, right? So what do? We give each unique YAC sequence a barcode, and then for each YAC sequence, we follow steps 3, 6, and 7. So we shear, we give each YAC big piece a barcode, we shear it, and we shear it again. Why? So that we can make them small, so that we can go to step 6, put the adapters on, and then 7, sequence it. Okay? So we have a big piece, but we still need to break it up into small pieces to sequence it. But we have this one big piece, we give it one barcode, and then we break it up. Because then why? We will be able to, we break it, then give it a barcode. And why? Because then we will be able to see this is that piece with this barcode. This other barcode is another big piece of DNA. This other barcode is another big piece of DNA. But do you see what this does? You have these big pieces of DNA that you need as the guide. You've broken them into small pieces, you've given them barcodes, so you can reconstruct the big pieces once they've been sequenced. Because they have each big piece has a unique barcode, right? So that's what you created. You created these big pieces. Then you bring them all together in the end, and you've recreated your big piece of DNA, right? So now, what do you have after 9 steps? You have DNA sequences from the same individual for different sizes of DNA. You've got millions of billions probably of tiny little pieces. That's from your first batch. You've got millions of medium pieces from your second batch. And you've got also millions of big pieces from your third batch. Actually, probably not millions, probably thousands of big pieces, right? So now you take the big pieces, and you see how they fit together, all right? And then you use the medium pieces. Where do they join onto, where do they line up onto the big pieces? And maybe they will also join some big pieces together. And then lastly, you bring all those billions of tiny little reads. Yeah? And now they're able to find the right place in the genome to line up. Okay? So, that is what you're seeing here. Here, you have each little gray bar is one 70 base pair Illumina read. And you see here how these are all stacked up on the genome. You see, and this, so for example, this red position here, you see it there? It's in that position is covered how many times on this genome? What is the coverage of this position here? It has been sequenced 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. It's not sequenced here, remember? But it's sequenced here, 16, 17, 18, 19, 20, 21. For some reason, it's not sequenced there. 21. So this site is covered 21 times. It has that site has a 20x coverage. So for that site, we'll easily be able to tell what is a heterozygote and what is a homozygote, okay? Because we've sequenced it so many times. Whereas, look at this site here in blue, okay? How many times has that site in the genome been covered by an Illumina sequence? Yeah? 1, 2, 3, 4, 5, 6, 7. Nah, that's quite low. Okay, you can be more or less confident about at least one of the base pairs, but you will not be able to tell which one, what is a heterozygote at that site. There's not enough sequencing. There's not enough coverage to determine what is a heterozygote at that blue site here, okay? But at this blue site here, and at this red site here, and at this red site here, there's plenty of coverage. You can call it heterozygotes, okay? That's what we mean by coverage, all right? So now look at this. You've got your big pieces, all right? You send them in first. You try to assemble them. Then you try to assemble your medium pieces. And then lastly your tiny pieces. Okay, that was your first batch, your second batch, your third batch. No? So the large fragments are a huge help because they can guide these medium and smaller on where to line up. Without these large fragments or medium fragments, it's very difficult to line these up. To make a de novo genome from just these tiny sequences is not possible. Okay, so that is how we make a reference genome. All right, guys, you will be asked about how to explain how you make a reference genome that are used to call heterozygotes. What do you know? That means that reference genome has to be at least 16 to 20x coverage, right? This is the method you use. All right, so this is another slide to show you the same thing. You see this big piece here? Okay, these big pieces are used as a guide to help the little pieces, the blue ones, to join. Okay, so that is how de novo genome assembly. So this is one of the most important tasks in biology, right? To assemble a genome when nothing else exists, okay? And if you don't have a good reference, many things are not possible, okay? You can't sequence second, third, fourth, and other genomes cheaply, for example, okay? And with the decreasing costs of the sequencing and the increasing demand for genomes and transcriptomes. Okay, this is bound to be a huge technology in the future. Okay, so this section is basically re-explaining to you how we do the assembly, right? If it's very complicated, if all we have are the billions of tiny fragments, okay? And that's why we need these DNAs of larger size, okay? The big ones, YAC and BAC libraries, barcodes, and the whole process again, okay? So the big sequences, okay, the clones of the big sequences can be used as a guide to assemble of the tiny ones, okay? And this is the whole hierarchical genome assembly process. You will be examined about this, okay? Don't memorize, understand. It's much easier. Okay, again, short reads, all the tiny reads, here are the long reads, okay? And here is the genome. You use these long ones to guide all these little ones on where they have to bind. All right. So the third generation, now next generation sequencing, is not just Illumina technology. That was the first kind, or one of the first kinds, sequencing tiny fragments. These guys at a place called PacBio in California have figured out how to sequence long reads without putting them into libraries. This is a huge cost to assembling a reference genome. You have to put each and every big piece into a YAC or BAC library, right? Imagine you don't have that cost. Imagine you can just sequence 2 million base pairs without PCR and without a library. That is what the PacBio technology is allowing us to do these days. So building a reference genome, the way we just learned it, in 5 years when you are doing your PhD, that will not be the way to assemble a reference genome anymore. You see how quickly the technology changes. Soon we will be using this PacBio technology to just sequence big pieces just like that, without PCR and without libraries, without YAC or BAC libraries. Okay? So this is just showing you how since 2005 is when NGS came out, Illumina has been the big technology until around 2013. In 2013, PacBio came and said, no, guys, we can sequence big pieces now using NGS, not just tiny ones that Illumina was able to do. And look, they've resolved bacterial genomes, the whole genome sequenced in one go. No libraries, no little pieces, no assembly, none of that. The whole sequence in one go. No? Then they did the yeast. Then they did 120 megabytes. Then they did 170 megabytes. Then they did a gigabase, sorry. That means how many in a giga? Huh? How many in a giga? Times 10 to the? Exactly. You got kilo, mega, giga. Okay? So, and then after that comes tera. Okay? So, you see the pieces of DNA that are being sequenced are getting bigger and bigger and bigger. So soon, we are going to be done with Illumina. So, just quickly to finish off the lecture, there are certain ways, after you've assembled your genome, right? There are ways to determine how good is that assembly. Is the quality of that de novo genome good enough? Okay? There are no real perfect ways, okay? But there are a couple of ways. And so the amount of sequence estimated, for example, the amount of sequence assembled should fit into the estimate for what is known for those species. So for example, if we try to assemble a human genome de novo and we get something which is 1 billion base pairs big. What is that telling you? That's telling you, Aish, but the human genome is 3 billion. That means we're missing 2 billion. That's not a good reference. Okay? Whereas if you're trying to do a bird genome, birds have smaller genomes. They have only 1 billion base pairs. So that 1 billion base pairs would be perfect for a bird genome. Okay? So how big is the assembly and how does it match up to what we expect for that species? How are the contig sizes distributed? What is the what is the N50 number? So the N50 number tells you, so contigs are basically pieces of the whole genome that are put together, okay? And the genome is made up of many of these contigs, thousands of these contigs, which are basically, if you can equate to the large pieces that we used for the assembly, okay? So how long is this contig is very important, okay? So the N50 length is the largest, is the largest number such that the combined length of all the contigs are at least 50% of the contigs are greater than this number. Okay, so the N50 number is basically an important way to say, okay, I've got a good genome assembly, or I've got a bad genome assembly. So if your N50 number is like, 30, that means that at least 50% of all your contigs are above 30 kilobases. Okay? So, and that's good. Whereas if your N50 was 2, then you're like, hey, that means 50% of my contigs are are bigger than 2. Only that means 50% of the contigs are smaller than 2. That means my genome assembly is not very good. The contigs are very small. So that's one way of figuring it out. Okay. But there is, as I say, no one metric that can tell you all the information about the genome. Okay? So the advantages of whole genome, well, do I really need to tell you? Do you really need to memorize what the advantage of having the whole genome is? You have everything. The whole genome. There's nothing more genetically that you can know. You got all the information you could possibly dream of having. That is the biggest advantage. Okay? And much more than GBS or RAD-seq. What are the disadvantages? Of course, you have a high error rate with Illumina sequencing. PacBio sequencing, the rate is a bit less, but they also have a high error rate, which means that you can't avoid high coverage. You must have a genome that have high coverage, where each place has been sequenced many times, so that you overcome this error rate. Okay? Because if you sequence the same base again and again and again, then you know it's not an error. It's actually the sequence that is there. All right? And of course, the high coverage is also to call heterozygotes. Don't forget that. To phase the genomes. Okay? And then short sequence reads are a problem. Right? But that disadvantage will hopefully go away in the next 5 to 10 years with new technology. So those short sequences are difficult to assemble. So actually, both of these problems will go away in the next few years, hopefully. So I know you can't ask me questions, but you are welcome to email me with questions, and we will continue the next lecture on bioinformatics. And that's the next big revolution in biology, because we had the NGS revolution, which created all this data. And now what do we do with all that data? We need to develop new softwares and computer skills to analyze the data. Okay? And that's what we call bioinformatics. And that's what we will talk about in the next lecture.